Good afternoon, everybody. So last time I asked who had been immunized. So does anyone know if they've taken antivirals? Tamiflu, Acyclovir. See, much fewer people than immunized, which is good. That's the way it should be. You should only take an antiviral if you're infected and you haven't had a vaccine. So that's what we're going to talk about today, antivirals. And as we said last time, vaccines have been pretty successful in preventing viral diseases. But if you're already infected, they usually don't have much of an effect, right? What is the one exception? You can be infected and still be immunized, vaccinated. Did somebody say rabies? Yes? Yes. Because the incubation time is so long for rabies that even after you're bitten, there is time to get the vaccine and to develop immunity that will protect you from dying, because rabies is about 100% fatal. Because the virus takes a long time to travel up nerves. But for all the other cases, when you're already infected, you need an antiviral. So that's what we're going to talk about today. That's our second arsenal, antiviral drugs. They can stop an infection once it has started. Now, we've been working on antivirals for about 50 years. I only have about 30. And part of the talk today, we're going to explore why that's so. And most of these are against HIV and herpes viruses. These, of course, not uncoincidentally, turn out to be chronic infections. They go on for long periods of time, so you can diagnose them and treat, as opposed to an acute infection, which is over in a few days. So here are some examples of some of the antivirals we have, not all of them. These are some targets here. You can see there are targets at many of the steps of the replication cycle: uncoding, replication, DNA polymerases and reverse transcriptases, processing, and virus release. And we'll talk about some of these in some detail today. And you can see mostly HIV, herpes viruses, influenza. We have 3 antivirals against flu. And coming up now are a number of hepatitis C virus antivirals, brand new ones, just in clinical trials about to be used. We'll talk about those as well. So why are there so few antiviral drugs? One of the reasons is that viruses are obligate parasites of the cell, so a compound that interferes with the growth of a virus may affect the cell. So you can have side effects often, and we don't want to have a lot of side effects, obviously. We want to minimize them. No antiviral is without side effects, though. So if you can find a specific virus function that doesn't exist in the cell, that's a good target, but there often is an overlap. Just a random overlap with a cellular function, so you have side effects. So that's one reason. Most of the compounds that are effective at inhibiting virus replication are toxic. The other reason is many of the important viruses are either very dangerous, or we can't grow them, or we can't test them in animals. So, for example, HBV, still no one can grow hepatitis B virus in a laboratory. Human papillomaviruses now can be propagated. It's not easy, though. It's a rather difficult system. For many diseases, there are no animal models. HIV, smallpox, measles. And these viruses are very dangerous to work with. Ebola, Lassa, smallpox. Yes? What are the reasons hepatitis B can't be grown? Why can't you grow it? I don't know. If we knew, we would grow it, right? People have been struggling for years. For Hep C, for example, from its discovery in the 80s, no one could grow it. And then, people made full-length DNA clones of the RNA genome. They put those in cells, couldn't get it to grow. And then a Japanese investigator found a different serotype, and that grew. So it was just a different sequence was able to grow. So now everyone is working with that serotype. With HPV, it required a specialized cell culture system, which was then developed, and you can now grow those. But HBV, still, no one can grow. And we don't know why. We don't know what the block is. So that makes it hard, but with recombinant DNA, the ability to express a single protein and look for inhibitors of its function, you can still develop antivirals, but they're hard to test if you can't grow the virus in cell culture. The first test, as you're going to see, is to see if your compound inhibits growth in cell culture. So that you have to bypass that. It's very difficult. Third reason is that any antiviral really must block viral replication completely. So we say it has to be potent. You can't have any room here for replication. Now, many of the standard drugs that we take, not for viruses or not antibiotics, but just drugs to treat various things, they work even if they partially block enzyme activity. Okay? But for an antiviral drug, you can't have partial inhibition because you will get resistance if you do that. So these are very different from most pharmaceuticals in that you require complete inhibition of virus replication. So they have to be safe, they can't have side effects, and they must be potent. So that's a difficult combination, and that's one of the reasons why we have so few. To achieve both, you have to do a lot of searching and chemistry and testing, and it takes a lot of time, and it's expensive. And as I said, if a drug doesn't block replication completely, we will get virus mutants. In fact, this is the perfect way to get drug-resistant mutants, is to have virus replication in the presence of an inhibitor. Even low levels of replication, because you will get viral mutants, and among those will be some that are resistant, and they will grow, and they will dominate the culture. If you have any replication, you can't get resistant mutants, because there's no replication at all. So you can have no selection. So this is a theoretical graph that just illustrates this idea. We have viral load here on the y-axis with time. So we have a virus infection, and we're looking at the amount of virus. We give a drug at a certain time. So in this system, we start out with high virus levels. We give drug, and we wait, and we measure virus over time. And the optimal dose of drug inhibits virus replication. So there's no more maintenance of virus right here, optimal dose. A low dose slightly inhibits, but eventually the virus rebounds because resistant mutants emerge. Same with even an intermediate dose. All right, so again, you can't have this middle-of-the-road inhibition. You have to completely inhibit. And again, that's different from a lot of other targets for drugs that we take, non-viral inhibitors. But you don't have to have complete inhibition to have a therapeutic effect. Yes? Right, so when viruses replicate, they make mutants, always. And so if you allow little viral replications in the presence of a drug, among the mutants that are made are going to be some that are resistant to the drug. And then those will replicate and take over, right? So you want no replication, zero, right? All right, so the problem of acute infections, that most virus infections are too short. By the time you feel ill, like in a common cold, it's too late to have an impact on the progress of the disease. Okay? So you have to give antivirals for these kinds of infections very early, and influenza is a good example. You have to take an antiviral for flu within 48 hours of feeling your first symptoms. Otherwise, it's not going to really affect the course of infection. Now, an alternative is to give drug to everybody all the time, right? It's not a good idea. Why isn't that a good idea? Yeah. Then if there's a little bit of virus around, you're going to select for mutants, and that's really bad. Plus, you don't want to give people drugs if they're healthy in general, right? Because they can't have side effects. And the more people you give a drug, the more likely you're going to see a side effect. So you don't want to give drugs to healthy people. If we had broad spectrum antivirals, this problem might be solved, because as soon as you feel ill, you take the broad spectrum. You don't have to diagnose what virus it is and get a virus-specific antiviral, right? But we don't have any such things. There's one hint of a broad spectrum against envelope viruses, but that's far from being developed. So right now, we have antivirals that are really specific for each virus. So you have to know what you have before you can be treated. And it takes time to identify the virus. So you feel ill, maybe you wait a day until you feel worse, right? And then you may go to a physician. Most of us don't. And then you have to get a prescription, then you have to go to a drugstore and get it. And this, by the time you do all that, unless you're very efficient, some people are, it's going to be too late. So there has to be some intervention here. And really the key, which I have mentioned before, is the lack of rapid diagnostic reagents. We could make antivirals basically for any virus that's around, but if you can't use them because of these acute issues, then they're not going to be made, and companies won't invest the money in them until we get rapid diagnostics. So one day we will have a rapid diagnostic that's very accurate. Right now we have some rapid ones for flu, but they're 50% wrong. Half the time they're wrong, so you might as well just take the antiviral when you feel bad with flu. And as I said before, that's the strategy that's taken. But someday we'll have better ones, and then you will see many more antivirals against acute infections. So let's go and look back at the history of antiviral drugs. The first searches really began in the early 50s. By this time, there were already a number of antibiotics used for bacteria, so people thought, well, we have these drugs that block bacterial infections. We should be able to do the same thing for viruses. And some of the early antibiotics were sulfonamides, so chemists began to modify these and check the derivatives to see if they would inhibit viruses. Then pox viruses were large targets back then because after World War II, smallpox was a big problem. In fact, as recently as 1967, millions of people died of smallpox globally. So there was still a need for antivirals against smallpox, and these are the thiosemicarbazones, which are found to be active. In the 60s and 70s, this got momentum, this whole idea of looking for antiviral drugs. Drug companies launched what we call blind screening programs to find chemicals that were antiviral. And again, this was spurred on by successes in treating bacterial infections. You know, in the 1960s, I think it was the Surgeon General who said, the end of bacterial infections is here. We have antibiotics, and we don't have to worry about them anymore. So it's 1960s. Now, of course, we have genes that make you resistant to all antibiotics. The New Delhi metalloproteinase makes all bacteria resistant to most of the antibiotics we have. It's pretty scary. So anyway, that spurred on antiviral blind screens. And what is a blind screen? You don't focus on a particular virus, but rather you just test random chemicals or natural products for the ability to block replication of a bunch of viruses in cell culture. Natural products are, you go out and you get some dirt from the woods and you look for something in it produced by a microorganism that will inhibit your particular virus. Companies still do this, in fact. It's a very profitable way to get antivirals because there are many, many microorganisms out there. So when you do these blind screens, you get what are called hits, something that inhibits your virus, compounds or mixtures that block your replication in culture. You then try and purify the active ingredient, and if you're lucky, you can synthesize it. You test it in various cell cultures and animal models. And then if you get leads from these kinds of assays, you then modify them by the chemists. So the chemists identify what the compounds are, and then they modify them to reduce toxicity, to make them more soluble, make them bioavailable so that when you take them, they go where they're supposed to, and also to improve their pharmacokinetic properties. You want them to last as long as possible between doses. So you can make chemical modifications that change all these properties. So these kinds of blind screens went through thousands and thousands of molecules before you even test something in humans. So you start with a hit, you make leads from it, you modify them, you make thousands of modifications in the chemistry lab, and then finally you find one which you think might be safe. You go into animal tests and eventually into humans. Despite all of this, from these blind screens, very little resulted. Now, one of the exceptions is called Symmetrel. This is the trade name for it. The chemical name is amantadine. This was approved in the late 1960s to treat influenza A viruses, and we still use it in certain situations. Most strains are resistant, but in some cases there is susceptibility, so it's still useful. We'll talk about the mechanism of that in a moment. But we didn't know what the mechanism was in the 60s when this was actually approved by the FDA. We didn't know the mechanism of inhibition. We just knew that it worked. Now we do know the mechanisms for most of the antivirals because, as you'll see, they're discovered in a way that makes you understand what the mechanism is. So the mechanism of amantadine inhibition was learned in the 90s. Today, it's very different. We use recombinant DNA technology to identify specific genes whose gene products can be inhibited. We use very sophisticated chemistry to modify molecules in any way that you want. We have a large database of knowing what kinds of modifications can be made to improve how long a drug lasts in you and so forth. So you can do very targeted discovery. We can clone genes, as I said. We can express them. We can purify them. We can crystallize them. We can see where they interact with their normal substrates and try and design inhibitors. As you've learned from this course, we know all the life cycles pretty well. So we can isolate a specific phase of the life cycle and say we want to make an inhibitor here. What are the enzymes involved? What are the viral enzymes that are involved? And so again, even for those viruses that you can't grow, you can get inhibitors. You can identify an enzyme, a protease, a nucleic acid enzyme, and look for inhibitors of it simply by cloning the gene and looking for inhibitors in cell culture. So no more blind screening procedures. You have a specific virus in mind, and you have a specific target. Just as an example, a few weeks ago, I called a large drug company because we wanted to write a grant to look for antivirals against dengue, which is a big problem, right? And this person that I reached, the head of outside interactions, said, oh, we only work on Hep C and HIV. So they're very focused. They don't want to go beyond their focus. No more blind screening. So if you look at a typical replication cycle, it gives you all of the possible targets. You have the virus itself. You have attachment and entry, translation, replication, genome replication in particular, and packaging and release. We do have inhibitors for many of these steps that we'll talk a little bit about. There are fusion inhibitors for HIV. There's a flu entry inhibitor. Interferon is a general inhibitor of translation, but it's really not a great drug. There are a lot of side effects. It's used mainly for Hep C. There are various inhibitors of nucleic acid replication for HIV and herpes, and there are even inhibitors of virus release. So we'll talk about some examples of each of these. But before we do that, just give you an idea of what it takes to discover a drug. This is the typical pathway to drug discovery. First you say, is the virus medically important? Okay, because if it's not, you don't want to spend money on it. So you identify a medical need. You do some research to figure out what parts of the virus you would like to target. So you identify a mechanism. So let's say you want to interfere with nucleic acid synthesis. It's a good bet because the viral enzymes are virus specific. You have to do a proof of principle. You have to somehow show that that gene is essential. And typically what's done nowadays is to delete the gene from the virus and show that the virus doesn't replicate. Because it could be that if you delete it and it does replicate, you could spend years and years in antiviral research on that target and it would be a waste. So then you start looking for inhibitors. You pick a target and you look for inhibitors. And again, the sources of these inhibitors, natural product libraries. Every company has a big collection of compounds they've accumulated over the years. You can make new compounds. You can now buy libraries. There are companies whose job is simply to furnish libraries of chemicals for various types of screens. And you go through a variety of screens. And we'll talk about mechanism-based and high-throughput screens. You get some hits. You then take those and from them develop lead compounds, which you can then modify. The chemists will modify that. Eventually, you can test them in animals and look at toxicity, pharmacology, metabolism, pharmacokinetics. And that is all related to bioavailability. Is the drug going to be where it's needed to inhibit virus replication? Once you're convinced that this is a good candidate, you can go into preclinical development, phase 1, 2, and 3 trials in humans. And eventually, if everything works out, you can get it approved by the FDA. But you cannot take dozens and dozens of candidates down this route. This is very expensive. So you have to make decisions about which ones to go with and which ones to cancel. Very tough things. So this process that we've talked about doesn't actually find drugs. A drug is a compound that's licensed, okay? So the end result in this pathway, a drug, is what happens after you do all of this. So this path of drug discovery doesn't find a drug until the very end. You get hits, you get leads, and you have to find a drug in the end out of all of that. Some of the things you need to answer in this process: will it get to the right place? Bioavailability. Will it persist long enough? Pharmacokinetics. And will it be safe? Toxicity and specificity. So only until you've answered all these questions do you have a drug that you can test in people. So we call this research and development for a reason, because you do research to find a lead compound, and then you have to develop it further, develop it so it's active, potent, not toxic, and so forth. And all of this can be $100 to $200 million per drug that you're testing. So that's why you can't do 10 drugs at a time. So if you're the director of antiviral research, you have to make a decision at some point along that discovery process. A person who used to work in the industry once told me those managers who canned projects usually lasted longer in the company than those who let projects go on longer. Because you can a project, you save money. Even if you don't get a drug in the end, you don't waste money. It's a little bit ironic, but that's the way it is. So you go through thousands of leads. In the end, you get one candidate for further development. And remember, you have to not only development is all the steps in taking the lead compound through safety testing, which is where a lot of drugs fail, scale up of synthesis. So you can have a very nice candidate which looks good in animals, then you go to scale it up and you can't do the chemistry, so it fails there. Formulation, how you mix it with other things to be taken as a pill or as an injection, pharmacokinetic studies, and then of course clinical trials, the true test, whether it helps people and whether it's not toxic in people. And these are the most expensive, of course, because they can involve hundreds and hundreds of people. 5 to 10 years typically after you get that initial lead, that is the first sense that a compound is going to inhibit your target until you get a drug. Now for HIV, this was fast-tracked because it was an urgent medical need, so this was cut in half really, and some drugs were out there in a couple of years. But for most drugs, it takes a long time and a lot of money. So here is the ladder of drug discovery. Now we start with hundreds of thousands of compounds in your library, and you do some screens, and you reject a bunch because they don't have any antiviral effect in cell culture, so the red is rejected. And then you reject more because they're toxic in cells. If they're toxic in cells, forget it. You don't even go further than that. Then you put them in animals, see if they have an antiviral effect, and then a bunch don't. You reject those. Toxicity in animals, you reject those. Then you go into people, and you reject them if they don't have antiviral effects. But in fact, here, we're not really testing 10 compounds. We're testing one. So it's either yes or no at this point. If it has an antiviral effect and it doesn't have toxicity, in fact, you would check the toxicity first and then the antiviral activity in humans. And then at the end, you have one compound after 5 or 10 years. So if you want to have a career in drug companies, this is what you're looking at. You can spend 5 or 10 years working on a project. Can you imagine then the FDA doesn't approve it? What that would do to your self-esteem? So you go to another company because you can't stand to see all the people you worked with through all those years. So here are some examples of screens that are done. This is a mechanism-based screen. We're looking for an inhibitor of a viral protease. So we know what the cleavage site is. We know what the protease is. So we have to do all the research to identify that. So here's our protease. We clone it and we can express it in some kind of system. And we develop an assay that's really quick and can be done on high scale. We have a bead attached to a short peptide that contains the cleavage site for the protease. And then we have a little green light here, which we can assay green fluorescent protein or something like that. And if the protease cleaves, it will separate the bead part from the soluble part, which has the green light on it. And you can do an assay. You can go time versus fluorescence intensity. And so you can separate, you can look at the supernatant and separate that from the bottom part here. And then if you have inhibitors, you can screen the inhibitors of the protease, because this line will be depressed if an inhibitor is present. So you measure the color in the supernatant, and then that's the way you can screen for an inhibitor. Here's another example of a screen. The previous one was in solution. There were no host cells. This is a cell-based screen where you take bacteria and you make them tetracycline resistant. for example, by putting a tetracycline efflux protein into their membrane. So now when you add tetracycline, the bacteria will replicate because they have this resistance pump that gets rid of the tetracycline. You can engineer a protease cleavage site for the HIV protease here. This is, in fact, one of the screens that was done. So now if you add protease to these bacteria, you can express it in them. It will cleave this part here, and it renders this efflux protein inactive. So now the bacteria are sensitive to tetracycline. So if you screen for inhibitors of the protease, those will make the bacteria tetracycline resistant. So that will be your readout, growth in the presence of a tetracycline for inhibitors that block the HIV protease. So that's a cell-based screen. Here's another one for a virus. Actually, this was just developed in the last couple of years. This is for Hendra virus, which is a particularly lethal paramyxovirus. It is active mainly in Southeast Asia, and it comes from bats and infects people. It hasn't gone from person to person yet, but it's extremely lethal. And one of the main ways that people get it, in Pakistan, is it Pakistan or Bangladesh, people collect date palm sap and they put it in these big containers and they leave it outside their house. And at night the bats come to drink the date palm sap because they like it. And they defecate and urinate in it, and they put virus in it. The bats have this virus, they're fine, and then people drink the sap and they get this infection. So we're looking for antivirals, but these viruses are very dangerous to work with. You don't want to be screening antivirals. So this was an assay actually developed at Cornell Medical College here in the city. What you do is you make a virus, you make a pseudotyped virus. You have, say, vesicular stomatitis virus, which is easy to work with and safe. And you engineer the glycoproteins of Hendra virus into it. So now it's not dangerous as Hendra would be, but you have the glycoproteins. And now you can infect cells and look for inhibitors of, say, entry, because now this is Hendra glycoproteins interacting with the Hendra receptor on the cell surface. But that only gives you one part of the replication cycle, just the entry step, because once the genome gets in, the particles that it produced don't have glycoproteins because you've actually deleted the glycoproteins from the viral gene, from the VSV genome here. These particles are made in another cell that expresses the viral glycoprotein, but the genes for them are not transmitted. What they've done here is they express the Hendra glycoproteins on the target cell. So now not only can this pseudotyped virus infect, but the particles that are released will have Hendra glycoproteins on them. They still have the genome of VSV, but you can look at the whole replicative cycle. You can look at not only entry, but budding from the host cell. You can get multi-cycle infection, and you can look for inhibitors of a broader range of targets. So this is being used now to look for inhibitors because Hendra is a longer acute infection, so if you diagnose it, you could apply an antiviral to save these people's lives. Now, high-throughput screening is a big part of drug discovery. You use mechanized approaches to do this. So you use multi-well plates. So many of you may be familiar with 6 well plates for growing cells in, or even 96 well plates. Well, this has 1,400 wells. They're very small. And in each well, you can do an assay for a different compound. And you don't have people putting compounds into these wells. It's too tedious and they would make mistakes. So you have these robots. You have rooms at companies full of these robotic arms. And what they're doing here is they're picking out the plates from an incubator and they're going to place them on a table. And then another robot will inject all the compounds into this and the arm will take it and put it back. So this is now how it's done by these robots, high throughput screening. So by this approach, you can do 10,000 compounds a day easily. You can rip through libraries very quickly, and you use chemical libraries, as I've said, that the company may have, or you could purchase them. Natural products, you go out. I know that Merck used to have a whole division of people that would go out on a daily basis and go to the swamps of New Jersey or the woods and just pick up samples and bring them back, and they would search them for active compounds. You can use chemistry to make thousands and thousands of variations on a compound. If you have the structure of your viral target, you can see where the natural ligand binds and design inhibitors that fit in there. We'll talk about an example of that. And now you can even do in silico screening. You use a computer to say what kind of compound will fit into this pocket the best. And you use the computer to iterate through many, many different kinds of structures, and it will tell you in general what kind of structure you need. Then you can synthesize it and test it. So that's called in silico screening. Now, when we make all these drugs, in the end, we're frustrated because we get resistance. And basically, any antiviral you make, you're going to get resistance to. You should not say if you're going to get resistance, but when. Because that's because viruses replicate very well. They have pretty good mutation frequencies, and we'll talk about that next time. Anywhere from modest to high mutation frequencies, depending on the virus. And this is a particular concern for viruses that replicate over long periods of time. It's paradoxical because these are the viruses that are best treated with antivirals. You can diagnose them and you have a long period of time for treatment. Yet because they're replicating so long, they're very likely to give off resistant mutants. We have mutants resistant to every drug in our antiviral arsenal right now. So we have, as I said, about 30 or so antivirals, and there are viruses resistant to every one of them. So and in fact, as you will see, it doesn't matter how many we make in the future, there will always be resistance to all of them. So you really have to have very special approaches to get around this problem. So right now, the antiviral arsenal is small. This has been overcome, as you'll see, for HIV. But we haven't done that for other viruses, infections. Now, what's the problem with drug resistance? This is really obvious. You can't keep treating with the same drug if a patient develops resistance to it. It's useless. Sometimes no other drug is available, so it's a death sentence for the patient. We do study resistance to understand how viruses become resistant. So we would like to know the sequences that are involved, and maybe we can get insight into getting around it. And maybe developing new strategies to get around the problem. What are the mechanisms? Well, RNA viruses, all nucleic acid polymerases, as you know, are error-prone. But those of RNA viruses are a particular problem because there's no correction mechanism. They can't correct the mistake they make. And they typically make one misincorporation for every 10,000 to 100,000 nucleotides that are polymerized. So remember, the RNA viruses have to replicate their genomes, and that's the error rate. It's about 1 million times greater than the error rate of our DNA polymerases. So that's quite high. So in an RNA virus genome of 10 kilobases, this range basically gives you one mutation in each genome or every 10 genomes, depending on the frequency. Yeah?
So I thought that with antivirals, it stopped replication. So how, I guess, why is there any RNA polymerase?
So why is there any replication at all? So it depends. If you start treating a person and they have a very large viral load, the likelihood is that there are already resistant mutants in there. That's just one scenario. The other scenario is that people stop taking their antivirals and you give the viruses a window to replicate. So in DNA viruses, the polymerase can excise and replace the mistakes. There are misincorporated nucleotides. These therefore evolve more slowly. So here's a DNA polymerase copying a template. Here's the 3' end of the growing strand. You have a misincorporation here, is shown as a mismatch. The wrong base was put in. It happens. But these DNA polymerases have 3' to 5' exonuclease activities, which can excise the mistake and replace it. So this makes the error frequency for DNA viruses much lower. It's not non-existent. And that's probably an evolutionary advantage. You should probably make some errors, otherwise you don't evolve, right? If you were totally genetically stable, you couldn't have any variation and you wouldn't be able to adapt. So there has to be some mutation rate. But this is why it's much lower for the DNA viruses. So let's talk about one drug in terms of how it works and resistance to it. Now, many antivirals are used to inhibit nucleic acid polymerases, and many of these compounds are nucleoside and nucleotide analogs. So here in the middle you have adenosine, guanosine, cytidine, and thymidine, the 4 bases that are used to nucleosides that are made to build DNA, and chemists have made derivatives of each of these that are inhibitors. And you can see all these compounds around the periphery, ribavirin, AZT, trifluridine. These are all derivatives of the 4 nucleosides that are inhibitory for DNA polymerization. We're going to talk a little bit about acyclovir, which you can see is a derivative of guanosine. You can see how it's different. It has the base, same base, but the sugar is incomplete. Okay? And this is highly inhibitory. It's a very effective antiviral. The way it works, it's used for herpes viruses. This compound, in order to be incorporated into DNA, that's how it inhibits, it has to be phosphorylated. And it is only phosphorylated by an enzyme produced by herpes simplex virus, the thymidine kinase, or the TK gene. So this is very specific for virus-infected cells. The phosphorylation won't occur. This first phosphorylation doesn't occur in uninfected cells. So you have the first phosphorylation event here. The P is added by HSV TK. So the drug gets into the cell. If it's infected with herpes simplex virus, it gets 1 phosphate. Then 2 other cellular kinases add 1 and 2 more phosphates. So now it's a triphosphate, and you need triphosphates to be incorporated into DNA, of course. Now this gets incorporated into DNA, right? So now we're putting in the acyclovir right here. But the problem is that you're missing the bottom part of the sugar, right? Which is where the next base would be attached. So remember, if this were guanosine, the hydroxyl would be the next site of the next triphosphate addition, but there's no guanosine here. So the chain incorporation stops. So that's why this inhibits DNA synthesis, because it's a chain terminator, we call it. Okay. We get readily resistance to acyclovir. These mutants arise spontaneously during virus replication. And there's some kinds of mutants that cannot phosphorylate the prodrug. These are mutations in the gene for the viral thymidine kinase. So they don't recognize the acyclovir. They can't phosphorylate, so it cannot inhibit viral replication. So these are variants of the TK gene that are selected for by the drug. Other mutants can't incorporate the phosphorylated drug. So with these mutants, the drug is phosphorylated. But there are mutations in the viral DNA polymerase such that they don't incorporate the phosphorylated acyclovir. Okay, so 2 kinds of resistant mutants. One, phosphorylation doesn't occur, and a second, the DNA polymerase doesn't use the phosphorylated compound. Now, this is a problem because you may not be able to treat the infected patient any longer. And this is a particular problem in AIDS patients who, as you know, are immunocompromised. And herpes infections can be problematic. So if you're treating an AIDS patient with acyclovir, you get resistance. You may get disseminated disease as a consequence. And sometimes you get cross-resistance. There are other nucleoside analogs, and you can try them, but depending on the mutation, it may be resistant to those as well. There is a different drug entirely called foscarnet, which is a DNA polymerase inhibitor. Sometimes the resistant mutants can be inhibited with that, but this has higher side effects than acyclovir. And if you are resistant to all of these, then you have no treatment options. So that's why it's important to have a big arsenal. And in this case, we don't. Now, acyclovir has been improved over the years. I just want to give you a sense for the kind of things that have been done. The original drug is acyclovir, and this has been modified chemically by adding a valine here, the amino acid valine. Then it's called valacyclovir. And this has improved bioavailability. So this can be an orally taken drug, and this one is more bioavailable after oral administration. What happens here, you take it orally, and then cellular enzymes cleave off the valine right here so that it can be phosphorylated. Otherwise, it couldn't be phosphorylated. And then this is the active form, so it's released in you. So this is then a prodrug, we call it. It's not actually the active form, but it has to be modified in you to become the active drug. All right, another one that we'll talk about is amantadine. That was one of the earliest antiviral drugs discovered. The target for this drug is the viral M2 protein, which I'm sure you all remember very well. This is an ion channel that is present in the virion. Now you remember the virion, which is shown here at the left, has glycoproteins, the hemagglutinin, the purple ones, the neuraminidase is the orange one. And then there's another membrane protein which is very low abundance in the virion. You can see it here. There's just one of them shown here. That is the M2 protein. It forms an ion channel. And just to review, during entry of the virus into cells, that's when that ion channel is necessary. So remember, the virus binds a receptor which has sialic acid in it, gets taken up by endocytosis. The endosomes get acidified. Protons are pumped into the endosome. That lowers the pH. And then as the pH drops, there's a fusion event that occurs between the viral membrane and the endosome so that the viral RNA can then get out and get into the nucleus. So you need low pH. Now during this step, as these protons go in the endosome, they then get pumped into the virus through this ion channel, this M2 ion channel. And you need to have reduced pH in the virion itself so that these RNAs can dissociate and go in the nucleus. So amantadine blocks the function of this channel. So it doesn't allow protons to go into the interior of the particle. Hence, when the fusion event occurs, these RNAs remain stuck to the membrane. They don't come off and they don't go in the nucleus. So that's how you inhibit replication. So here is a model of this. The M2 ion channel is shown here. It's a tetramer. We're showing two chains here. This is the lipid membrane of the virus. So normally protons would be pumped through here, but the amantadine fits right in the channel. It blocks the protons from being pumped in. So that's why it blocks infectivity, because it blocks entry or uncoating of the viral RNA. So in fact, by studying amantadine and amantadine resistance, we learned a lot about uncoating. We didn't know that acidification of the virion was needed for virus uncoating until we started studying these amantadine-resistant mutants. Because we had amantadine, we knew it worked, we knew it blocked infectivity, we didn't know how, and then people got amantadine-resistant mutants, they sequenced them, and they said, whoa, the mutations are in M2. That's unusual. So what is M2 doing? And that led to discovering this. So really, that's why studying resistance can be really important. Another flu antiviral is an inhibitor of the neuraminidase. So we haven't talked much about this protein except to say that it cleaves sialic acid from proteins on the cell surface. So let's talk about its function. And when flu viruses infect cells, remember new viruses are produced by budding. All right, so viruses are produced by budding. These viruses get glycoproteins from the plasma membrane, the HA and the NA, and then the released particle floats away from the cell. Now, there's a little paradox inherent in this, in that, if you remember, the receptor for these viruses is sialic acid, and sialic acid is present on this cell. So as these new viruses are budding off, they would just stick to the surface of the cell if they didn't have neuraminidase there to clear the sialic acid off of the places where the viruses are budding. So in fact, many years ago, it was shown that if you block the function of the viral neuraminidase, the viruses that are produced all cluster on the cell surface. So this was done in the 1960s. And you could see the viruses never got away. And so that led to the idea of inhibiting the neuraminidase. Maybe that would be a good antiviral approach. Now, many, many years later, the structure of the neuraminidase was solved in conjunction with sialic acid. So the substrate for the neuraminidase is sialic acid. This is sialic acid right here. It fits into a pocket. And what the neuraminidase does is it cleaves it off the protein that it's bound to. So the crystallographers looked at this and they said, let's design compounds that look just like sialic acid and make them bind tightly in the neuraminidase so that they block its activity. They prevent sialic acid from getting in. And so that's how the two neuraminidase inhibitors that we use today, Tamiflu and Relenza, that's how they were developed over a 30-year period. So they're the two compounds, zanamivir or Relenza, and also oseltamivir or Tamiflu. All right? And these were designed to mimic sialic acid, which is the ligand of the inhibitor. And the idea was, if we make these look really much like sialic acid, the virus will not be able to mutate to resistance, because then they won't be able to bind sialic acid, and then the viruses will be dead. So this would be a good strategy, okay? So they did that, but only one of these two is the really good mimic of sialic acid, and that's zanamivir. Oseltamivir is close, but not exactly like sialic acid. So guess which inhibitor you get resistance to? The one that's exactly like sialic acid, or the one that's not exactly like? You get resistance to Tamiflu. It's a lot of Tamiflu resistance. So here's a cartoon that illustrates this. So here is neuraminidase on the virion surface and sialic acid on a glycoprotein on the host cell. All right? So remember, what's happening is the neuraminidase is binding the sialic acid, and it's going to cleave it off so that the virus particles can get released from the cell. Now, oseltamivir and zanamivir fit into the pocket that would normally be occupied by sialic acid. And as I said, zanamivir fits really just like sialic acid. You can see that we've drawn it like a triangle here to mimic sialic acid. But oseltamivir is slightly different. It deforms the pocket a little bit. It's not really like sialic acid exactly. So you can get mutations in the neuraminidase that block the binding of Tamiflu. But these viruses are still viable because they can still bind sialic acid. But we haven't seen many mutations conferring resistance to zanamivir. Some, but not as much as Tamiflu. Here's some numbers for you. So there's a really nice CDC website where you can get weekly information on influenza virus isolates in the US and their resistance and so forth. So the top one is a summary of resistance among influenza viruses worldwide as of December last year. So these are influenza A viruses. There are two subtypes of influenza A circulating, H1N1 and H3N2. You can see both are resistant to amantadines. Those are the M2 channel inhibitors. So amantadine's pretty useless here. Amantadines don't work against influenza B viruses. They don't have an M2 ion channel. And so far, all the isolates are susceptible. The majority of isolates are susceptible to Tamiflu and zanamivir. Now, this is the new pandemic strain that emerged in 2009. Before that virus emerged, there was another H1N1 strain circulating. It had almost 100% resistance to Tamiflu because we'd been using it for 5 or 6 years. So over that time, we got all Tamiflu-resistant viruses. This is the situation as of October. So let's look at influenza A isolates. There are 586 isolates that they tested, and they have one virus resistant to Tamiflu. So that's not bad, but there is one out there, and when there's one, there's going to be more. And then we have the pandemic H1N1 strain, 2,298, 17, which is 0.7%, so slightly higher than H3N2. So it's not a lot, but the number is growing. And with each successive season, the more we use these inhibitors, the more we select for resistance. So eventually, all the isolates will be resistant to Tamiflu, most likely. I wanted to go over one other inhibitor with you, and this is an inhibitor of picornavirus uncoding, which was discovered in 1985. And I like the first sentence of this article here. This was done up in Rensselaer, New York. Any fans of Rensselaer here? No? The discovery of antiviral drugs known today can be attributed primarily to the tenacity of researchers and the serendipity of circumstances. This is a good summary of antiviral discovery back then. It's changed a lot now, and it's a lot more focused. But look, several years ago, a program was initiated to identify compounds which demonstrated antiviral activity against any of a number of RNA or DNA viruses. First compound to show activity was the diketone shown down here. So they had a big chemical collection. They were just screening for inhibitors. They found this one that worked against a rhinovirus. Following this discovery, several structural modifications were made to examine the following parameters. So they made all these derivatives of the original compound. The chemists went in and added side chains or rings and so forth. And they simply looked at the minimal inhibitory concentration in micrograms per mil. And you can see some of them were not active at all, some of them were more active than others, and they eventually picked the best one and went forward with it. And this was the genesis of these Win compounds. I think I've told you about them before. These compounds bind into the capsid of picornaviruses in a hydrophobic pocket just below where the receptor binds. And here is the structure of one of these Win compounds binding in this pore. It fits tightly in this pore and prevents the virus from undergoing conformational changes that it needs to undergo to uncoat. So this is an uncoating inhibitor. Unfortunately, this never made it to be approved because resistance emerges very readily with the use of these compounds. It was tested for rhinovirus activity, but there's so much resistance, and the rhinovirus colders are such short duration that it never made it to market. But these are used in the lab to study the process of uncoating. There are many new targets that are being explored. There's, for example, the hepatitis C virus protein shown at the top is a polyprotein. It's made as a precursor. It's then cleaved by 3 different kinds of proteases. There's a viral serine protease here. There's a viral metalloprotease here. And then these cleavages are done by a host signal protease, the host signal peptidase. Would that be a good target for developing an antiviral, you think? The host signal peptidase? No. Probably have really bad side effects because you need that to cleave signal sequences from proteins. And this one was shown very early on to be essential. If you deleted this protease from the viral genome, it wouldn't replicate. So this is now the target of antiviral drug discovery for hep C. On the bottom is another protease from a different virus. This is the herpes virus cytomegalovirus. This is a very unusual protease structurally. There doesn't seem to be anything like it in the cell. So the idea would be that if you could find a specific inhibitor, it might not have side effects. But again, you don't know until you put it into animals. There are clinical trials ongoing for the hep C drugs. I wanted to show you. You can find these announcements very easily online at various websites. And these are 2 companies starting a phase 2 trial, larger numbers of people answering different questions of a protease inhibitor. And also there are polymerase inhibitors for hep C being developed as well. And these are oral hep C drugs. There's also mRNA antivirals that are being tested. MicroRNAs are short 22 nucleotide RNAs that bind target sequences and cause them to be inhibited in a variety of ways. We've talked about translational inhibition mediated by microRNAs. And in this experiment, they took the hepatitis C virus RNA and designed specific microRNAs to match sequences in the genome. And these inhibit virus replication. So for example, here is an assay for virus replication. This is a northern blot type of assay. You can see here the viral RNA is replicating quite nicely. If you add a microRNA against GFP, it doesn't affect viral RNA levels. But if you add a virus-specific microRNA, you block virus replication. So the microRNA, again, binds to the viral genome and inhibits it. And these are being used or being considered for clinical trial as well because these can be delivered in a way that makes them quite stable. So microRNA targets for some viruses. HIV antiviral discovery is the most extensive of any. There are many targets for intervention of HIV replication, including attachment, reverse transcription, integration, transcription and post-transcriptional processing and assembly. And we're going to go through a couple of these as well. The problem with AIDS is that you have years and years of virus replication. Remember, 10 to the 10th particles a day are produced. And we'll look at the numbers, but that's almost impossible to get around. The first HIV drug was AZT, which is a thymidine derivative. And the way it works is that it's phosphorylated. AZT is taken up by the cell. It's phosphorylated by 3 cellular kinases, so there's no restriction to virus-infected cells as there is for herpes simplex virus. And then the triphosphate drug is incorporated into DNA and causes chain termination because there's no hydroxyl here. There's an azido group. That's the Z and the AZT. So it's chain terminator. Initially discovered as an anti-tumor compound, actually, and then was checked for HIV inhibition, and it worked, and is a chain terminator. The reason it is somewhat specific is that it's not a great substrate for cellular polymerases. It's a little better for the reverse transcriptase. So it's somewhat selective, but there are many, many side effects of AZT because it does inhibit cellular polymerases. So unlike acyclovir, it has substantial side effects. The other problems, it's taken orally, it's got good bioavailability, it's absorbed rapidly, but the half-life is horrible, an hour. So you have to give doses 2 to 3 times a day. And this is problematic. It makes you feel lousy. It causes anemia. There are many side effects. And this kind of regimen, short half-life with multiple dosing, is a good way to select for virus mutants. And in fact, that happened immediately after the drug was initially licensed. Mutants resistant to the drug arose. These contain single amino acid changes at various places in the reverse transcriptase so that the drug, the phosphorylated drug, would not be incorporated into the DNA. So the altered enzymes don't bind the phosphorylated drugs. As a result, new analogs were developed, chemical modifications of various sorts or modifications of other nucleosides, and some of those are listed here. And that allowed double combination therapy to be used, the use of 2 antivirals at a time. Nevertheless, within a year, even using 2 drugs, double resistance arose. So this led to the development of non-nucleoside inhibitors of RT. So these are drugs or compounds that don't mimic nucleosides, but rather they fit into the reverse transcriptase and they block its activity. So here's the structure of reverse transcriptase. The polymerase active site is right here, and these non-nucleoside inhibitors bind a short distance from the active site. So they probably perturb the active site enough so that it doesn't work and inhibits the enzyme. And there are a couple of these that are being used at the moment. Now also, you get resistance to these, of course. Very rapidly, amino acid substitutions in the enzyme that prevent the drug from binding or modify the structure of the enzyme even when the drug is present. So you can't use these by themselves, and they're typically used in combination therapy. Now when you have so many drugs that are available, you can then see if resistance to one drug does something with respect to resistance to another. And you want to know this because you have a patient that becomes resistant to one drug. You don't want to give them another drug to which they're already resistant. So you need to know their resistance profiles. And some surprising results have emerged. And these are all examples of where resistance to one drug actually restores susceptibility to another. So here we have, for example, a change which confers resistance to this inhibitor. And then if you put them on a second drug, didanosine, and you get resistance to that, it restores susceptibility to the original drug. So this is the case for all of these compounds. The resistance to a second compound restores susceptibility. Yeah.
So does that mean that if you were putting someone on heart, that you would try to use drugs which have like this seesaw thing going on?
Well, in heart there are only 6 combinations that are approved, and these are some of the considerations that are part of that. Yeah, you want to make sure that you're not going to get, if resistant to one doesn't give you resistance to all 3, but also some of these considerations come into play as well. So actually you don't make up your own combinations, you have to use pre-approved ones, and those issues have come into play. In fact, nowadays if you are diagnosed with AIDS, you have to have a resistance profile done. The best way is to sequence the genome in all the areas of known resistance and see what mutations you have, and then pick the treatment based on the sequence. And obviously the more sensitive the sequencing, the better off you are, because there can be very small variants present which might not be detected unless you're doing very sensitive sequencing. There are a number of anti-HIV drugs that target the protease. Remember, the virus matures by assembly of a polyprotein that's processed by viral proteases, and the virus buds at the surface. One of these proteins is the protease, PR, the yellow circle here. It's incorporated into the particle and processes the polyprotein as the particle matures. And very early on it was shown that this protease was essential, so it was used to develop antiviral drugs. A key discovery was that it would recognize small peptides. You didn't need a whole protein substrate to be recognized, but you could have a small peptide, and that The active site of the protease contains 7 amino acids of the substrate, so that was also useful. So initially, a number of inhibitors, protease inhibitors, that had already been investigated in hypertension studies provided the first leads for this work. These protease inhibitors actually inhibited the HIV protease. These are peptidomimics. They mimic the natural cleavage site. So these were the first drugs, and then subsequently mechanism-based screens and other structure-based designs were used to discover more. So here is the sequence of the protease substrate. These are the amino acids that comprise one cleavage site in one of the viral proteins, and this is the recognition sequence here. And then this inhibitor on the bottom mimics this protease cleavage site. You can see here, and then it's different on either side. So this binds very tightly to the protease. It's a peptidomimetic. It mimics the substrate and inhibits its activity. So now we use combination therapy, triple therapy, because the resistance, the probability of resistance is just the product of the individual resistances. So if you have 2 drugs with probabilities of resistance 1 in 10 to the 3, 1 in 10 to the 4, you need to have 10 to the 7 virions to get resistance, which is not a lot considering you get 10 to the 7th, 10 to the 10th particles made a day. And typically you target different mechanisms in this cocktail. And as we've said before, that way you can treat the infection as a chronic disease. And very recently, one pill was approved that has all 3 inhibitors in it, which makes it much easier than having to remember all the combinations on a daily basis. So here are the mathematics of drug resistance, which are worth going through. If you assume that 1 mutation is needed for drug resistance, which sometimes is true, but not always, and if the virus mutates 1 in every 10 to the 4 bases, which is once per genome per cycle, then in every 10,000 viruses, every base is substituted. So any pool of 10,000 viruses that you put out has a change. In each virus, there's a change at a different position in theory. So you make 10 to the 10th new viruses a day, so that means 10 to the 6th viruses are produced each day with resistance to 1 drug, 1 million. That's why 1 drug doesn't work very well. Now, if you take 2 drugs, 10 to the 8th viruses, and it's divided 10 to the 10th over 10 to the 8th, 100 viruses are made each day that are resistant to both drugs. So that's a little better, but eventually you will get resistance in about a year. And then if you do 3 drugs, the rate is 10 to the 12th. So you need fewer viruses, but eventually you will get there as well, depending on how much the growth is going on. Remember, replication is suppressed, so the number of 10 to the 10th particles a day is going to be brought down significantly by triple therapy, so it will take much, much longer to get resistance. My understanding is that you do get resistance, but they are mostly a consequence of people stopping their therapy for whatever reason, and the virus grows and the resistant mutants grow back. Some new antivirals have been developed recently. One of them is the integrase inhibitor that we have mentioned a couple of times in this course. Remember, the viral DNA integrates into the host DNA, and this integrase protein mediates the strand invasion by the viral DNA. The integrase inhibitors simply bind the integrase and prevent this joining, this nicking and joining reaction. And this is an example of one of the integrase inhibitors. You can see it's a small molecule that forms a variety of interactions with specific amino acids of the integrase. Now, of course, you can get resistance to these as well. And those have been well described in the literature. You can see here integrase inhibitor resistance mutations. There are 2 different integrase inhibitors shown at the bottom here, and all the different amino acid changes that you can get to give resistance. I thought I would show you this study, which is one way that you test an antiviral drug in a clinical trial. So this is a double-blinded, randomized, placebo-controlled trial, a dose-ranging study, different doses to find out which one is good. And they put 200, 400, and 600 milligrams of raltegravir and, of course, the placebo. And these are blinded, so nobody knows who's getting what. And then they measure the viral RNA either the change in baseline or the proportion of patients with RNAs of different copies per mil, less than 400 or less than 50. So this is the gold standard, getting less than 50 copies per mil. And then the change in the CD4 cell count. So you can see these are the numbers for the placebos, and if you compare the drug treatment, let's take the highest amount of drug, 600 milligrams, you can see that this is a big change, a negative change in the baseline of RNA compared to the placebo. Proportion of patients with less than 50 copies per mil, 66% compared to 13%. So you can see this is quite effective, slightly more effective than 400 milligrams per mil, 400 milligrams. This is an inhibitor of CCR5, which we talked about a long time ago. CCR5 is the co-receptor, remember, that is needed for HIV infection. It's a small molecule that binds in CCR5 and causes a change in the molecule such that the virus doesn't undergo high affinity binding. But of course, you get resistance to that as well. And finally, there are fusion inhibitors. These are actually peptides, 36 amino acid peptides, which interfere with the fusion reaction. So normally, when fusion occurs, here's the viral membrane and here's the host cell target. GP120, or the envelope protein, binds the receptor on the host cell. The fusion protein is exposed, inserts into the cell membrane, and then remember there's a conformational change in this protein. It hairpins, draws the two membranes together so that they can fuse. We looked at movies of this a long time ago. These peptides, what they do is they bind these alpha helices that have extended here in the glycoprotein. So these small cylinders are the peptides, the antiviral peptides that are binding, and they lock it into this position so it can't undergo the hairpinning and the fusion. So these block fusion. These are very expensive drugs, and they have to be reconstituted just before injection. So you get them as a dry powder, you add buffer to them, and you inject them. And it's very expensive. And of course, we get resistance by amino acid changes along these alpha helices that simply prevent the binding of the peptide. And some of these are just examples of the kinds of mutations that you get. These have been well studied. Changes in GP41, which is the subunit of the glycoprotein to which these peptides bind. And these are all tracked at a website, the HIV-DB, where you can look at all the resistance mutations in real time. So this is the chart of all the available HIV antivirals at the moment. They're classified according to the mechanism. So you can see nucleoside RT inhibitors, non-nucleoside inhibitors, protease inhibitors. Then we have our fusion inhibitor. We just talked about the CCR5 inhibitor, integrase inhibitor, and then various combination products. So this is the tripla, which is the pill that contains 3 different drugs, and then other combinations can be given separately. What's interesting about this table is that you can see the very, very rapid approval rate for most of these drugs. It usually takes much, much longer to have drugs approved. So this is the time to approval where you submit your data to the FDA and they say it's okay. And this, of course, being an urgent situation, they're all fast-tracked and given fast-track type approval. So some of them are just a few months to approval, which is amazing. So you see we have a big arsenal, and that allows it to be effectively treated. Yes?
You might note that there's now quadrivalent guideline.
Is it, sorry?
There's a quadrivalent.
4 drugs, yeah?
Yeah, Gilead has phase 3.
Okay. And what would be the resistance to 4 drugs? Be very high, but eventually you'd probably get there. But remember, this is why a lot of genomes on the planet, and no matter what you make, you're going to get resistance.